first up, it's going to be Valerie Bentivegna, working for the Akamatsu lab, is going to be talking to you a little bit about a multidisciplinary approach to studying actin dynamics and endocytosis. I have no idea what any of that means, and so I'm as excited as you are.
Thank you so much, Andy. And don't worry, I will explain all of the words that you can see on the slide. So yeah, my name is Valerie. I'm a research scientist and also lab manager. I work in Akamatsu's lab. We're a relatively new lab, started in June 2022. So it's been really exciting to be part of a growing lab. And I just want to talk a little bit today about what I do in the lab, what the lab does as a whole, and kind of how we approach doing science together. And just as a teaser, I have this really fun GIF that you've seen running the whole time. So this is a side of a colony of stem cells. And you can see all these little blinking lights coming in and out. And every single time you see that, what you're seeing is an endocytic event. Again, I'll explain in a second what that all means. First, a little bit more about myself. Hard to put yourself on one slide, but I tried. I have a background in cell and tissue scale biophysics. I swear this meme was relevant when I made it, probably 10 years ago. I like finding creative solutions to problems, extra bonus if they have Lego. You can ask me about it later, but this was how I used Lego in my PhD research, which I thought was cool. And this here is a Lego microscope that I use to tell K-8 students how optics work. And then finally, I am a science comedian and a storyteller, usually with this guy here. That's Bruno. He's a blue ukulele, and there's more stickers on him right now. Anyway, so a little bit about me, and then basically what does the Akamatsu lab do? We focus on the mechanical relationship between the actin cytoskeleton and mammalian endocytosis. Let me start with endocytosis. Endocytosis is the process that cells use to take things, basically eat stuff in their environment. So as you can see in this cartoon right here, there's this little fold in the membrane. It's the membrane folding in on itself and creating a little vesicle to take something out from the environment. This is how viruses can get into our body. This is how the cell recycles membrane proteins. It can whatever's floating around here kind of goes into the cell. So it's a very important process for a lot of different things that the cell needs to do. And then actin are these red fibers here. Actin are cytoskeletal elements. They are these long fibers that form. They're important for cell migration, for cell shape, for all these different things, including endocytosis. And they have a mechanical force. So to be able to do this process, there has to be some force pulling the membrane in. And that is exactly what we study in the Akamatsu lab. So we're trying to look at how does the actin cytoskeleton self-assemble at these spots of endocytosis? How does the actin cytoskeleton adapt to different forces in changing environments for different cell types to make sure that these processes can happen? So we're at one lab and we take multiple different approaches to study this. One thing we do is computational modeling. Unfortunately, this video won't play, but if it would play, you'd see all these little fibers coming in. Basically, these white fibers are the actin. And then this is the endocytic pit. And you would see it kind of pulling it in. And we can use computational modeling to look at things that we won't be able to see if we try to do light microscopy because it's just too small. We do stem cell tissue culture and genome editing. What you're seeing here at the start is a single cell growing into a colony of stem cells, and it's moving really quickly. We do some 3D printing. This is mostly because I'm a huge nerd and like to do this. But we also can print proteins so we can see kind of how they fit together, how they self-assemble. I also print a lot of tube racks just because they're handy in the lab. We use molecular tools. What I'm showing here is from a digital droplet PCR readout. We do some PCR. We do immunofluorescence staining. We do a lot of different things using molecular tools. And we do fluorescence microscopy. I don't know if you can see this, but again, they're a little, looks like little blinking lights happening. This again is a colony of stem cells that are doing endocytosis. And so we're kind of using those tools to study closer how it all works. What's interesting about our lab, you can see we do a ton of different things. We have a ton of different members that are coming from different backgrounds. We have someone who is mostly doing computational modeling at the moment. We have someone with a stem cell background. I come from biophysics and 3D cell culture. How do we kind of bring it all together and organize our research as a lab and make sure it's coherent? I think it's quite unique. We use a tool called Roam Research. It's a flexible note-taking platform. I use it on the web. And everything is interlinked with each other. So I'm showing my daily notes when I log in the morning. This is what shows up, and it tells me, oh, these are what I plan to do today. If I click on that link, I can see what everyone else was planning to do that day, which is kind of cool. Sometimes you can see who's planning it to be in the lab. And every single bullet here is called a node. So every single thing is kind of a link on its own that you can link from other places. That sounds quite abstract, but we use it to kind of map out how different ideas are linked together. For example, to answer a scientific question, we can use this tool to break our inquiry into different parts. For example, question, claims, and evidence. I'm going to use the example for this of how bendy is actin. Making that sound a little bit more scientific is what is the persistent length of bending actin filaments? So that is our question. It's its own node. And we can link that to a piece of evidence that we found in a paper. That could be a figure that kind of shows what we're trying to find. And we kind of make an evidence statement just summarizing what that finding was. So that piece of evidence is informing that question, and then we can make a claim. That's kind of a more general statement from, we found this in the literature. We can have different pieces of evidence supporting that claim. We kind of like hold this to be, at least for now, as far as we know, the ground truth. And like, all of this is based on sources when we're doing this in a literature research system. And similarly, we can break projects and experiments into kind of its atomic parts. Discourse graph system is really great because it supports being a transparent lab. We have multiple different lab members. We're all taking notes in the same place, which I think is quite cool because everything is shared, everything is linked. I can go see like, oh, my lab member already did research on this and they already found this perfect figure that's relevant for it. I can go ask them about it. I can just read what they already wrote about it without having to like go dig in a notebook. So what you're seeing here is all these gray lines are links between these different nodes that were created. We use this tool to do basically everything we do in our lab, literature study, experiments. We track our daily notes in there. We track our meeting notes in there, etc. I also like this tool because it kind of supports our lab value of having accessible and open science. It's an easy entry point for new lab members. Not only do we have all our tutorials and like protocols on there, you can also just jump into a scientific question no matter what your level is and kind of see what already exists in our lab's knowledge about that. And because it's so structured, you can kind of just already bullet out a piece of text and then write that into a narrative, and then you have a manuscript, which is quite cool if you want to publish something. So with kind of like how we approach taking notes and sharing along in our lab, I just wanted to show one example of a bit of research that I did. So I said my background was in biophysics. I worked on 3D tissue models. And part of that work, so this is from my grad school thesis, which I never thought I could put in a PowerPoint here. But I learned that different cell types form different 3D structures when we embed them in extracellular matrix gels. The extracellular matrix is the stuff that kind of sits between your cells and gives your tissue structure. We can also use it if instead of growing a cell on a flat piece of plastic or glass, you can put a cell in the middle of extracellular matrix and it forms these little 3D structures. They kind of form balls. Depending on the cell type, they can be solid balls or hollow balls. And these here, we call them luminoids because they have a lumen in the middle, which is quite cool. And they're very useful because you can study cell types that have like a clear polarization. There's an apical side and a basal side. You can study all these different systems in an environment that's a little bit more native than you would if you're looking into 2D cell culture. And the question is, what about the cells that we use in our lab? What happens to them if I put them in an extracellular matrix gel? What happens to them is that they form luminoids. So they form these hollow structures. On the left, you're seeing a phase image. You can kind of see like a donut shape That's how they show up in this imaging modality. So they're hollow in the middle, and you can see a single cell layer. I need to click play for a second. I wanted to skip ahead. Come on. So it's not letting me skip ahead, but you can kind of see in fluorescence microscopy that it does form this round structure that kind of looks like a soccer ball. It's hollow here in the middle. We don't quite know it's in the middle of there, but you can again see kind of endocytic events happening now in this 3D system. Great. I'm going to build up on this slide kind of an overview that the way I built it in Rome Research. It's a different view that we can use. Basically, I was working on a project growing luminoids from these stem cells. I did a couple of different experiments. I had a bunch of results. I can create these nodes with the results, and it's cool because they show up with a nice little picture. I can make a ton of claims about it, and then those claims can lead me to ask further questions. Now that I have this model system, what can we do with it? What does this even mean for the lab that I currently work in? For example, how are there differences in endocytosis depending on cell location? We can look at in the middle versus on the cell membranes versus on the outside. Do we see a difference in dynamics of endocytosis? Things like that. Also, if I vary the stiffness of the extracellular matrix shell, how does that change how endocytosis works? So it's been really nice kind of forming this new model system that we can now use in the lab to explore the questions that we're already asking in 2D, but now in 3D. And with that, I wanted to end on one last slide. I work in the Akamatsu lab. A little over a year ago, we were 3 members. Now we are 7. And it's been really fun to see the lab grow. So definitely thank you to everyone. We do a lot of fun things. We don't just do science. We also go to escape rooms and play board games. And when not everyone's there, we make sure that they are there in spirit. So yeah, thank you to everyone in my lab who's made this a really fun place to expand my enthusiasm for doing 1,000 different things at the same time. Also, thank you especially to the Terrio lab who's made being a lab manager really easy. The top 4, we're on the top, we have the best view, no dispute, and obviously the whole department. So yeah, any questions?
We can, of course, start off with any of the new recruits if you guys have any questions at all. And after that, of course, we have our own questions, and last but not least,
Go ahead. What were you using the Legos? Okay, so the question was for the Zoom, what I was using the Legos for in science. So this is going to take too long. I will not go back. I was doing ultrasound on these 3D structures, and when I just grew them on a plastic plate and like kind of floating around, ultrasound bounced too hard from the glass, so it was really hard to actually see what was happening. So I created, using a different material, kind of a mold, and used the Lego wells was like my little chamber. So it was smaller, more confined, also less reflective, and I can actually see what I wanted to see. And then the cool thing was, at first I was trying to like sand off the Lego, but then I realized I should leave it because I could use it as like a marker to see, oh, if I'm looking down a microscope, that's where something was. And now in ultrasound, I can find the same spot. So they gave me a lot of Lego when I graduated. Uh-huh. Rome Research isn't. There's a free similar type of thing called Obsidian, which is the same type of note taking. Everything's bidirectionally linked, and I use it for my personal notes right now because Google Keep is not great, and it's a lot easier. So I don't know if they have academic discounts, but feel free to talk to me or Matt for more details. about the Gospel of Rome and obsidian.
How are you wrestling your new lab members awesome results and all
It's kind of happened really organically. So we have an undergraduate student who's been really great at like making sure when we take really bad notes that they get better, and that all the context is there that we need. By example, I guess, like showing you can structure a whole experiment like this, and that means that you can at a glance just be like, "Oh, right, that's what I did. Cool." And then because it's so great, it convinces people to do it. Yeah, exactly. So, thank you very much.
And we will go ahead and get our next speaker ready. We're going to kind of zoom out on scale a little bit, moving away from cell biology and getting into probably our largest zoom level. It's going to be ecology with Katie Holt working for the Boersma Lab. Whoa. With feeding penguins, sex but not size related to foraging success of Magellanic penguins. Go on ahead, Katie.
Thank you so much. Let's see if I can flip this on. Thank you. We're going to take a really hard left turn. I'm sorry, keeping you on your toes. Welcome to the University of Washington and to the biology department. My name is Katie. I'm a graduate student here in Dr. D. Boersma's lab. And today I'm going to be sharing one of my findings that sex, but not body size of individual Magellanic penguins is related to their foraging success. But before I get into that, I sort of wanted to give some context as